Thank you, Prof. Felicia. I'll be very, very short. And I begin by thanking you for asking me to be with you today for this 7th annual Rare Disease Colloquium, which, from what I understand, is expected to be more of a scientific session about the research going on rare diseases and on discussions on available new technologies and new drugs and treatments that could become available. It's my pleasure to make available this hall, which since the mid-17th century served as the stables of the Grand Master's Palace. This place was enlarged in the first half of the 17th century by Grand Master Antoine de Paul and continued to serve as the residence of the subsequent Grand Masters, civil commissioners, governors, governors generals, and also presidents till this very day. I felt it my duty to welcome you and wish you a fruitful discussion for this colloquium. As you may know, I come from a medical background, but I have not touched the subject in my initial remarks, knowing how specialized the subject is. What I can safely say is that rare diseases, because of their low incidence, are often, as you very well know, referred to as orphan diseases, because they attract relatively little interest from researchers and worse still from pharmaceutical companies who would rather invest in research of a condition which is quite diffuse, like cancer, and hope to find a silver bullet drug that would make all shareholders rich. From the ethical point of view, that is not good. However, the situation is what it is. Even though I have to note that the rare diseases have been given due consideration recently, very recently, in April, only 2 months ago, by the European Commission in its proposal for an ambitious overhaul of pharmaceutical legislation. In this proposed reform, special incentives are being provided for medicines addressing significant unmet medical needs of rare diseases in the form of an increase to the standard duration of market exclusivity, plus the possibility of benefiting from additional periods if they launch the drug in all member states or develop new therapeutic medications for an already authorized orphan medicine. The other known fact is that such medicines, if they become available, will be very costly. And I'm glad to know that locally, treatment for one of these diseases, cystic fibrosis, has been provided free by our health system since a few months. In today's world, the medical sciences have progressed beyond belief, aided as they are by developments in biomedical technology and better experimental tools, such as molecular biology, genomics, and such other examples. Hopefully, understanding the basic pathology, the deficiency or metabolism, or the physiological reasons behind these rare diseases is always a step forward in finding drugs or finding ways of, say, genetic manipulation to avoid these conditions or to bring them back to normal once they develop. On the last note of thanks to Professor Felice, who has been a pioneer here in Malta in the field of rare diseases. I wish you all fruitful discussions.
Thank you, Your Excellency. And we now move forward with the scientific program. And I assure you that you will be hearing sound words from many who are committed to the cause of rare disease research and development. So we should start with our friend, Professor Ruben Cauchi, who's a colleague in the Faculty of Medicine. Ruben is associate professor with special interest in neurogenetics, and he leads Malta's national ALS registry that includes a specialized biobank or collection. And his goal is to identify new genetic and environmental risk factors, because it may not be all genetic. I mean, there is this possibility of complications from that which is genetic and that which is toxic, environmentally toxic. So, Ruben, the title of Ruben's talk is Decoding ALS in Malta, Flying Towards a Better Future. What I wanted to say in the beginning, that today's overall topic is about new developments, by which we intended new tools. And one of Ruben's tools are the flies these very special flies that he will talk about.
Thank you. All right.
Okay. So, okay. First and foremost, I wish to thank the organizers for their kind invitation. I can say that I'm really excited to be here today because I can give you a flavor of our research program, which is directed at understanding the cause of ALS. This, we believe, is the first step towards discovery of urgently required effective treatments, allowing us to guarantee, therefore, a better future to ALS patients in Malta and worldwide. So, let's begin. What is ALS? ALS stands for amyotrophic lateral sclerosis, and it occurs when there is degeneration or death of the motor neurons. Motor neurons, we have two types of motor neurons in our body: the lower motor neurons, which make direct contact with the muscles, and therefore they control the muscles that we use to talk, eat, breathe, and walk. And the lower motor neurons are themselves controlled by the upper motor neurons, which are going to relay signals from a region in the brain known as the motor cortex. Now, what causes ALS? This is a big question. We don't know that much, but what we know is that we have these so-called established or undisputed ALS risk factors. And these are male age, an increasing age, having an athletic profile, and most importantly, a very good dose of bad genetics. So, back a few years, we wanted to understand ALS in Malta, and therefore we set up a national ALS registry and biobank with the aim of capturing the epidemiology of the disease on the Maltese islands, and importantly, identify those risk factors that are specific to Malta and Cauchi. So we can say that we're privileged to visit every single newly diagnosed patient at his or her home, where, subject to written informed consent, we collect a blood sample, as well as data on environmental and lifestyle exposures. Back in the lab, we extract the DNA from the blood samples, bank the DNA, but most importantly, we analyze the DNA to check for differences, the so-called genetic variants between ALS patients and healthy elderly volunteers, which we use as controls. And interestingly enough, the first big discovery, surprising discovery of our work was that the incidence rate of ALS in Malta is actually one of the highest in Europe, and actually well above the European average. And this, we think, in our case, in the case of Malta, our evidence is telling us that genetics is driving the disease, and that's possible because of genetic insularity. Obviously, that comes from our isolated geographical setting. Now, a certain thing that we have learned from our journey is that the cause of ALS is actually different in different patients. And today, we give one single drug to ALS patients. Only one drug has been approved. And because of the vast differences in the cause of ALS, the clinical efficacy of the drug that we use is actually extremely low. So we do envisage a future whereby we're able to stratify patients into subgroups, allowing us to give drugs that target the specific cause of ALS. And that, we believe, will improve substantially the clinical efficacy of such drugs. And since gene-targeted technologies, gene-targeted therapeutics, actually are more effective when given at disease onset or at a pre-symptomatic stage, nowadays, genetic screening technologies are increasingly an integral part of our healthcare. Actually, we do envisage a tomorrow whereby our children will be governed by an algorithm that is able to predict ALS risk. And if the risk is high, we can devise preventative measures, and we can give drugs at an early pre-symptomatic stage. Now, in Europe, mutations in 4 genes cause more than 60% of ALS cases, more than 60% of the ALS cases in Europe are actually explained by mutations in these 4 genes, the so-called big 4 genes. So it's not a surprise that molecular therapies targeting these genes are at an advanced clinical stage of development. And in fact, in the case of one gene, the SOD1, one therapeutic actually reached FDA approval a few months ago. Interestingly enough, from when we analyze the genetics of Maltese ALS patients, we find only a few patients that had mutations in these big 4 European genes. And actually in Malta, we have our own big 4 genes, which are different from the European genes. And we do find several patients with mutations in the so-called ALS2, DAO, SETX, and SPG11 genes. Anyway, these are interesting genes for us, for our population. The problem is that these are genes which are infrequently damaged in Europeans, and therefore they remain a very low priority for drug development. And that is why in my lab, we are studying these genes. We have a vigorous research program focused on these genes with the aim of identifying drugs that will be mostly beneficial for Maltese ALS patients. And to do so, we make use of the fruit fly model system. Why is that? That is because of the high degree of genetic and biological conservation between us humans and flies. Here we have a model organism that is able to age rapidly. So we can model a neurodegenerative disease of mid-age very well. And importantly, we have a highly accessible neuromuscular system. So as a proof of concept, we have studied the DCTN1 gene, which encodes for the dynectin protein. The dynectin protein is a protein important for the transport of molecules along axons. Axons are those long, thin cables of motor neurons. And this is a gene which we find damaged in a female ALS patient living in Sliema. When we switched off this gene in the neurons of flies, we found that flies became increasingly paralyzed, and this is the same as happens in ALS patients. When we delved deeper and looked at the contacts that motor neurons make with the muscles, the so-called neuromuscular junctions, we see that the neuromuscular junctions of those flies which have loss of the dynectin gene, they are small compared to the healthy controls. And again, this was similar to what we see in ALS patients post-mortem. When we looked at the molecular landscape, we identified several genes that are incorrectly spliced, incorrectly edited. And in particular, we identified a gene target, ANC13, which we can rapidly activate in the flies to decrease neuronal death. And in so doing, we can rescue the ALS symptoms in flies and hopefully one day in ALS patients. So that's all I have to tell you today. What I want to say is that this work is not mine only. It's a team effort of a lot of enthusiastic PhD and MSc students and postdocs. So big thanks to them. In addition to that, a big thanks goes to the patients and healthy elderly volunteers that took part in our studies, our collaborators at Mater Dei Hospital and at international universities, and obviously various funding agencies that have supported us and keep supporting us to do this work. So thank you all for your attention.